Good afternoon, ladies and gentlemen. Thank you very much for your attendance in this session. This is the last session of a three-day conference. So I think it's been exciting, but also probably you have been a bit tired, but I try to make yourself enjoy the session. The session title of today over here is Post-Pandemic Global Clinical Trial, Trends in Drug Development, Innovation, Analysis, Execution. And also we are going to talk about changes and adoption in COVID-19. We are going to have 5 speakers, including me. So we are going to have a bit busy session. And I'm going to run the session with 5 presentations without Q&A and taking Q&A in the last 10 minutes. Just because the session is a bit busy, I would like to ask each speaker to keep the time, 20 minutes. Okay, I'm going to introduce myself. My name is Han-Lim Moon. I'm a medical oncologist by training, and I'm working in pharmaceutical industry sector as a pharma R&D consultant. But also I'm supporting pharma and biotech companies to develop their compounds at the clinical development stage. Today, I'm going to talk about what is the success for the drug development. Sorry. Yeah, so I'm going to talk about overarching strategy of drug development for global success. And as I said before, through pandemic, there have been quite a lot of changes in terms of drug development, but some of those essentials would have not been changed. So that the spirit should be maintained as it was and then as it is. This is the outline I'm going to talk about. When we are talking about global success in drug development, we have to define what is global. Is it conceptual or is it territorial? Actually, if you think of regulatory bodies who approve the drug, there should not be any party who represent global. So I'm rather going to talk about US FDA point of view in many cases, just because they have very transparent and then they're very clear guidance. But also US market is very big, or biggest, so that if you are able to make a success in US NDA, probably you can pass the first toll to make a great success. However, we also think about global success, including other territories as well. So European territory, very importantly, Northeast Asian territory, including China, Japan, Korea, and Taiwan. Of course, other countries are important, but for us, probably these are important territories. In addition, if you work with the IND, not limited to NDA, you also have to think of territorial strategy for IND. So which territorial countries you would like to go? Of course, US very important if we aim to have NDA in US. But also Korea, of course, very important as well. But if you have any ambition to get approval in China, Japan, you should probably open the trial in those territories. I used to work for GSK as the Northeast Asia strategy leader. And Asian strategy in GSK was considered as a very important, just because the Asian economy was getting bigger, but also Asian disease sometimes different from others. I'm a medical oncologist, as I said before, so that I specialized myself into anti-cancer drug development. And then if you see, some specific cancer disease in this territory includes gastric cancer, hepatocellular carcinoma, biliary tree cancer, and then different type of non-small cell lung cancer, i.e. EGFR mutated cancers. And China and Japan strategy, very important. just because not only their economy is very big, but also they have very specific regulatory requirement. So for China, they need to have China-specific trial. In Japan, the same. They should have their own population phase 1 trial to see the PK, if Japanese PK is similar to others. Therefore, if you think of Asian strategy, you really have to be very serious about China and Japan strategy. China previously, it was a bit difficult to go. A lot of their own cultures, some difficulties hidden without any clarification. But these days, since 2016, they made a big NMPA reform, so that their regulation is a lot clearer than before. So I think it's relatively easier than before. Japan's strategy, PMDA, pretty clear, but also they have very difficult part of regulation demanding Japan-specific data. If we are talking about success in drug development, there should be a lot of different definitions for the success. But the most important definition is actually approval by the regulators, which is the permission for physicians to prescribe the drug in clinical practice. And then an NDA success rate is, as you know, pretty limited. So 1 in 20 to 1 in 10 would be successful. So typically in oncology, it's more difficult, but overall average, we would say 10%, which means if you develop the drug, you have 10 compounds, probably 1 would have drug approval. So actually, everybody dreams of approval, but actually it's not very easy objectives. And also you need to see what environmental change is like. I'm going to talk about Manny's definition later on, but if you want to develop drugs, you need to think of what are the current standard of care in different diseases. And if you think of many drugs under development and then approval almost every week or every other day, you should think of what is dynamic change of standard of care. So in 2021, FDA approved 16 new molecules and then 50 subsequent indications. That means almost 1 or 2 indications were approved by FDA, which actually means standard of care has been changed once or twice a week in some disease, some cancer disease. I'm going to talk about Keytruda success story. I think some of you already know, but it's very, very interesting story. The reason why I would like to explain the Keytruda success story is there are a lot of lessons, how to do and what to do to make a great success. Keytruda, as you know, is a PD-1 antibody targeting cancer immune suppression. So it will release immunosuppression. But originally, the scientists wanted to develop PD-1 agonist to treat autoimmune disease. He was not successful, but finally he's got Keytruda. At the time, it's pembrolizumab or some compound name. And when he made it in Organon, he also had to experience 2 occasions of M&A. So Organon was acquired by Schering-Plough. Schering-Plough was acquired by MSD, so that the scientists and then the developers continuously had difficulties and frustration, because company continuously wanted to deprioritize the development. But finally, when MSD looked at success of ipilimumab, they decided to go for IND. And one luck or the good success case was when it went to the clinical development, one of the melanoma specialists has seen 7 out of 10 patients responded to the drug. So he was not able to sleep if the company wanted to discontinue the development. So at every case he has seen the response, he continuously sent it to MSD. So asked the developers to raise it to the upper management. And also, they made the team continuously bigger, but one of the important contributor was Dr. Roger Permuter, who actually devoted himself to Keytruda development. He talked to everybody in MSD, you should stop what you are doing. You should do this work, i.e. Keytruda development. And also escalate to the upper management. We shouldn't have any upper limit for the budget. So his passion and his effort has made indeed great success of the Keytruda success. And also, one of the investigators from Hopkins, they did MSI high indication, and then it has made a great success as well. So from this lesson, it has shown that how scientists, clinicians, and then companies' effort would make the success as a collective intelligence and also collective effort. And then one of the important story later on, one patient who had got colorectal cancer in his 20s and then metastatic disease in his 30s. So he was waiting for hospice care, but finally he's got Keytruda, and then later on he got to live very long, and then he married, he had another life. So this is the drug development success. This is the very complicated study design and change of Keytruda. Pembrolizumab, Keytruda, at first, they started as a very small scale dose escalation phase 1 study, but later on, they changed 8 times of protocol amendment. They amended protocol 8 times, and finally, they ended up with having 12,060 patients in phase 1. And then they were successful to get 2 indications through that trial, one melanoma, the other was non-small cell lung cancer. And as you can see, in 2022, they will have above 20 billion as one compound. But do you want to repeat the same story? So this is a well-known story of sintilimab by Eli Lilly and then Innovent. They did exactly that same study as Keytruda did in non-small cell lung cancer first line. The data is more or less similar or very similar, but US FDA rejected to approve in many reasons. So in a way, drug development, if you want to repeat the same story, sometimes it would work, sometimes it would not, just because environment is changing. And in global success, you should make a very good strategy in the beginning, but also you should make a very precise clinical development plan, and then also clinical trial design, and then execution is also very important. And at the end of the day, in many countries, health outcome is also very important to get reimbursement. So all of these essential components should be combined together to make a great success. I'm going to talk about a bit unmet needs. Many times, many people talk about unmet needs. Oh, lung cancer is highly unmet needs. But do they really know lung cancer is composed of more than 10 different molecular types, and then many different histological types, and then stage 1, 2, 3, 4, or first-line metastatic, second-line metastatic, third-line metastatic. So you should really know what is unmet needs. Unmet needs means there is no drug at all, no approved drug, or if there is some drug, but still the drug needs to be improved just because the drug is not safe enough, not convenient enough, not efficacious enough. But again, if any drug is approved in one disease, but it is accelerated through accelerated approval from US FDA's point of view. Actually, in US standard, it is not considered as available therapy. That means if there are some drugs approved by FDA through accelerated approval, actually still the disease belongs to unmet needs. NDA is through the review for CMC, clinical efficacy and labeling. And then the efficacy should be considered as substantially better than enough than standard of care. And then what is the meaning of substantial benefit? Substantial benefit is not only about statistically significant data, but also clinically meaningful data. And what are the definition of efficacy or benefit? It's through clinical endpoint. So if the patient feels better, if the patient functions better, or if the patients live longer. So you should have one of these endpoints and then also suffice enough through clinical trial. And then through regular approval, it should be phase 3 or randomized phase 2 or a big enough size of a single phase 2 trial. And FDA, actually, as I said before, the assessment of significance of clinical efficacy is really about clinically meaningful data. I said there are a lot of changes in clinical standard of care, and this is lung cancer field. As you can see, so many drugs has been approved the last 10 years. So more than 15 drugs in different settings, metastatic setting, adjuvant or neoadjuvant setting. And then also there are a lot of immune-oncology drugs in the second line, first line, and then also neoadjuvant or adjuvant setting, so that you need to think what is the true unmet needs continuously looking at the changing environment. If you really want to make a drug successful enough, actually you should know where to go. So you should think of what are the data through phase 3, then you think of phase 2, and then you should think of phase 1 to make a success at the end, phase 3. So actually, it's kind of backward thinking how you define and then how you make a success through the clinical development. And also, indication prioritization, especially in oncology, is very important. Usually, the entry indication is through very rare disease or rare indication. Traditionally, actually, many pharma companies develop the big indications like lung cancer, breast cancer. But nowadays, many times, they enter the market with small indication, but extend their indication later on. And phase 2 and phase 3 strategy, normally phase 3 is for regular approval. Phase 2, more about accelerated approval. But you need to think which strategy is better to get actual NDA approval. Phase 1 strategy is also very important, especially in oncology. What first in human dose you are taking, and then what kind of dose escalation scheme you are taking as well, and then how you define recommended phase 2 dose. Because sometimes MTD may be the phase 2 dose, but many times it's not the case, probably biologically optimal dose. So you should think of what are the best dose for phase 2. NDA strategy, do you want to take a regular approval, or do you want to go through the accelerated approval process? Nowadays, FDA is more conscious of accelerated approval through single-arm trial has shown many failure in phase 3. So they recommend to have randomized trial even in early phase development. So randomized phase 2 after 30 to 40 patients data. However, still FDA highly recommends to think of accelerated approval, especially for the cases of high unmet needs disease, i.e. oncology or some rare disease. So you can think of strategy, what to do for the expedited program, but also reasonable enough clinical trial design. Market access and then health outcome, very important. So you should implement your health outcome data inside the trial, many times nowadays, even in early phase trial. So my talk has been very long, but I can summarize a few important things. When you develop the drug, you should think of what is the final goal. So how you make a success as through NDA. And then you should make a whole strategy as a whole before you go to individual trial. But once you make a clinical development plan, you should make a very precise clinical trial design. And speed and quality, very, very important. And then also you should think of health outcome data. But at the end of the day, another important thing is what regulatory strategy do you have? So do you want to go through accelerated approval, or is it enough, or you want to go to regular approval? So the drug, which is valuable to go to the patients, it should have good enough data, but also the speed and development, very important to arrive to the physicians, and then finally to the patients. Thank you very much. Okay, I'm going to introduce the next speaker. Next speaker is Dr. Daegon Kim, who is the CEO of Voronoi in R&D department. He is specialized in developing targeted agent. He previously worked in KIDI and MIF, and now his team is very strong to make a lot of good small molecule. And today, Dr. Kim's topic is improving tolerability of targeted anti-cancer drugs through innovation of drug discovery. So over to you. Thank you.
Thank you, Dr. Moon. 네, 저는 이번 코비드-19 기간 2년 동안
During the COVID crisis for the past 2 years, there have been several innovations in drug discovery.
개발하는 쪽에서 다들 관심을 가지고 있습니다.
And I believe that as a person who are engaged in new drug development, I think this is truly noteworthy, and I would like to take this opportunity to introduce that to you. When it comes to the drug discovery, we were not really affected by COVID crisis. For DD for license out, we received these data remotely, and we had more video conference. I think these are the only changes that we experienced after the COVID. And in terms of the regulatory processes, there was one important change. And also through the innovative technologies, we were able to successfully overcome that. So I would like to share that with you. Protein kinase or PKC data inhibitor and various PKC substances, they have been utilized for blood cancer. But because of the malignancy issues, FDA announced that the clinical trial should be stopped. So that happened during the COVID crisis. And that was one of the most important incidents in oncology clinical trial. So that's why I think Dr. Moon emphasized the importance of dose optimization for oncology clinical trial. What FDA announced at the time was that, so when they give them the accelerated approval, you see that the hazard ratio was pretty low, but then when they looked at the overall survival rate, the combination therapy actually reduced the survival period of patients. That's why they stopped the clinical trial. And eventually, and also what's important is safety margin. In other words, within the therapeutic range, how much safety margin we can secure. And the clinicians like the Dr. Moon, during the clinical development, they claimed that the dose optimization is not fully taken into consideration for clinical design. But for a person like me, who are mainly engaged in drug discovery, our concern is more with, like, if the drug does not have the selectivity to distinguish normal cell and cancer cell, then the drug will have anyway some fundamental limitations. So in terms of discovery, how can we overcome this? And this is regarding the Afghanistan aid during the Obama administration. And here you can see, so let's say that the cancer cell asked the normal cell that, are you okay? So we have to tell the difference between these two. PI3 kinase-based blood cancer drugs do have some issues. And last year, Relay and Oxon Oncology, these two companies, they came up with very interesting pipeline. Here you can see the protein kinase 3K alpha and PI3K alpha. It has some hotspot mutations in various cancers. So one of them is in the regulatory domain. Liver cancer, breast cancer, and colon cancer with great unmet needs. You can see that this kind of mutation are pretty prevalent in these cancers with a lot of unmet needs. And if you look at the cycle of the occurrence of this mutation, as for the A7047R, it's just next to the active site, and regulatory domains are pretty apart. So the red part indicates that's something that requires our attention. So ATP or the substrate binding site. But you can see that the other substances are somehow attached and change the structure. So this is how Relay approached this. And for this hotspot mutation, as for the direct inhibitor, Oxon Oncology came up with some solution for that. And this is an example of breast cancer, especially the hormone-positive breast cancer. More than 40% of the patients show the mutation of the PI3K alpha. And as for the hotspot mutations, more than 60% of the patients do have such mutations. In other words, 1 out of every 4 patients do have the PI3K alpha mutation, and that actually led to the breast cancer outbreak. And also, as for the hormone-positive breast cancer, usually they receive the hormone therapy plus the chemotherapy, but during the chemotherapy, they can apply the targeted therapy. So we can create some market for that. And if this market can successfully be created, then it will give huge opportunities for industry expansion. And so here you can see the drug developments in relation with PI3K alpha. But one of the most difficulty is that it is an important modulator that delivers the signal to the insulin inhibitor. And so when it is activated, then the plasma glucose will go inside the cell and produce the synthesis for glycogen. And therefore, this will increase the blood glucose level, which will again release more insulin, and which can aggravate the diabetes. And for the PI3K mTOR, in the beginning, the inhibitors, they had a very serious toxicity. And regardless of the PI3K alpha subtypes, you can see that most of the inhibitors show very serious toxicity. And even the alpelisib that was selected for PI3K alpha inhibitor only, and it has some PI3K alpha benefit, but because of the limited index, although they received the approval, but they had a lot of difficulty in dose optimization clinical trial. And as for the mutant-specific PI3K alpha inhibitor is currently under development. And so I think within this year, 2 product pipelines will be available for clinical trials. And For the epilepsy, you can see that the biggest concern is with the hyperglycemia. And for all grade patients, here you can see, and most of the patients show the grade 3 or grade 4. So now you have a question whether this drug is really a drug or whether it's a toxic agent because it caused a lot of AEs. And not, aside from the alpelisib, another drug is now under approval process. And Relay, they can target the 3 mutations and selectively inhibit the mutation. As for the OXO drug, they are selective only to a certain type of the mutation. And also the sparing is about 90%. Currently, they are in non-clinical phases. So currently, the toxicity and efficacy are now being studied. But in terms of the hypoglycemia or high insulinemia, these kind of AEs were not observed at all in relation with diabetes. Here you can see the Loxo product. So this is a PI3K inhibitor. And you can see that there are about 30 times sparing. And as for the cell proliferation, you can see that about 90-fold sparing, and they effectively target the wild-type mutation. And during the AACR, the relay shows the potent mutant isoform selective biochemical inhibition. And so wild type is about 15 nano, but as for the mutant, it's about 5 nano on average. So in other words, 10-fold sparing effect. And from the Loxo-Genographic model, you can see that the Loxo achieved a significant tumor regression in breast cancer xenograft patients. And also in terms of the C-peptide and the plasma insulin, there were no differences, whereas the alpelisib, you can see that there were some AEs in relation with diabetes. And in Relay, this shows the 10-fold sparing effect. But for the xenograft patient, when we look at the safety level, you can see that they are in the normal range. And also DRF toxicity study shows that about 90% of the ICP plasma concentration is achieved. And even in this situation, there were a stable level of insulin. So the insulin level was normal, and glucose level was normal as well. So alpelisib, although they got the approval, once these drugs are entering into the clinical phases, I believe the alpelisib will be very hard to be sold. And now here you can see this is the 90-fold sparing. And versus the 10-fold sparing that target the 3 all mutations. So this is a Relay product. But then all of a sudden they talked about the KRAS. So for a KRAS inhibitor, there is a GCM-specific inhibitor, and GCMC and V, these subtypes, different mutation subtypes can also be found. So the RLI claim that the drug that targets all 3 mutation is more effective than the other counterparts, but we are not sure whether it's really effective. And also the other types of the mutations can happen anytime. So what's important is that in the kinase domain, so there is a drug that binds to the active site, but there are also some drug substances that bind to a site that's very different from there. So this will expand the options for patients. If the patients take one kind of drug and experience problem, then they can switch to different drugs. So I think that this could be a good news for many breast cancer patients. And as for the PI3K, I would like to give you 2 independent examples. They are not related to each other, but both of them occurred during the COVID crisis period. So I would like to share with you these 2 examples. And also, for the past 2 years, there was a very interesting announcement from the AACR. So this is about the PRMT5 cooperative inhibitor. This is a little bit unknown drug, and it was announced by AACR. As you can see, P16 is a well-known tumor suppressor, and oftentimes in tumor cells they are deleted. And just next to it is the MTAP gene, and MTAP is it degrades NTA to arginine and methionine. So it's a hydrolysis agent. And these two genes are located very closely, and so it could lead to various collateral damages. So CDKN2A, if there is a mutation, then oftentimes co-deletion could occur. So two of them could be deleted together, and they actually represent about 15% of the solid cancer MTAP deletion prevalence. So once MTAP is deleted, then the MTAP cannot be hydrolyzed, and therefore the MTA will be accumulated in the cancer cell. And with the accumulation of MTA and methionine release, but if the methionine decrease, then SAM will also decrease, and increase in MTA will bind to the PRF5, and therefore inhibiting the activation of the enzyme. So this will create the negative feedback. And SDMA and RNA splicing, these are essential substances for the functioning of the cells, but there will be shortage of that. And PRMT5 will have some problems with this deletion. And therefore, if you inhibit the PRMT5, then the cancer cell will be damaged before normal cells. That's the basic approach that they take. And here, one more interesting technology that I'll let you share with you is this. So general PRMT5 inhibitor, this is a GSK material developed for PRMT5 inhibitor. And you can see the therapeutic window is just too small, and therefore they stopped the development. And cooperative inhibitor here, like the AMG 193 and Tango, and also the Miratis substances, they're about to be studied in clinical trials soon. So I'd like to briefly talk about the concept of these new substances. PRMT5, they deliver the methyl base to create essential substances, but if with the MTAP deletion, then SAM will not be released smoothly. Then MTAP substrate and MTAP will proliferate, and then they will bind to the PRF5 and work as a negative inhibitor. And as for the MTAP, they will just bind to the external outer envelope. But as for the GSK substance, they create the substances that bind to this area and to the inhibition. Then as for the cooperative inhibitor, you can see that these normal cells that bind to SAM or PRMT5, they actually inhibit the binding into the PRMT5. And they also have some interactions with the MTAP, and therefore with MTAP deletion, and if MTA proliferate, and also many of them are combined to the PRMT5, only for these types of the cancer cells, they can have some effects. So this is a pretty innovative technology, very outstanding. We were all surprised by this technology. This is a very mature, I think, and very innovative technology, I guess. And here you can see that so for normal cells, it has no effect, and it only targets cancer cells and lead to the cell necrosis. So for cells with and without MTAP deletion, you can see some differences in terms of activation. As for the GSK substance, where the clinical trial is stopped, there is almost no margin, but somehow they were brave enough to start the clinical trial. But the other substances, you can see that efficacy is pretty strong. And in xenograft and PDX model, again, the model speaks for themselves and prove the efficacy. So if these two substances enter into the clinical phases, I'm sure that this will provide great benefits to various types of patients. And as for the MET2A inhibitor, because time constraint, I'm going to skip that. But as for the MET2A inhibitor, together with the cooperative inhibitor, it doesn't have the cancer selectivity, but it increased the therapeutic window with the high specificity. So if it's combined with the MET2A inhibitor, then it can lead to some synergistic effects. And therefore, MET2AI is not in competition with the PRMT5. Rather, it's a sort of the synergistic relations with this. That's why the pharmaceutical companies have accelerated the development of MET2A inhibitor. And in terms of the drug discovery during the COVID crisis, there was no significant changes. But during the period of COVID, the several companies have come up with some innovative pipeline. And what became more clear is that we have to create the cancer-specific substances. And also, as for the cancer metabolism targets, we don't really have drugs. So between the substrate and end product, if there is a negative feedback between them, and because of that, the structural differences can be created. And so if we could develop a drug that can distinguish such structural development, this will, I think, lead to a better and more significant innovation. With that, I would like to conclude my presentation. Thank you very much.
Yeah, thank you very much, Dr. Kim. I'm going to introduce Dr. Kim Eun-kyung, who is the Executive VP in ABL Bio. She is in charge of clinical development, including strategy and management of the studies. Dr. Kim was working for Novartis and BMS previously as the oncology development lead. And Dr. Kim is a certified pediatric oncologist, and she has long experience in clinical development. Today, Dr. Kim's topic is how important the speed of drug development is. Thank you very much. Yeah.
네, 소개 감사드립니다. ABL 바이오에서 근무하고 있는 김은경.
Thank you very much for the wonderful introduction. I am Kim Eun-kyung, as was introduced, from ABL Bio. This is my disclaimer. It says that my statement is not representing the company's views in ABL Bio. So the views expressed are my own. And this presentation is for academic discussion purposes only. And the author, I, do not intend to promote any products described herein. As was asked, I was about to talk about the importance of the speed of the drug development. Most of you already agree that that is very important. And my topic today is how important that is for the speed. If you look at the history, the fastest one that has been developed was the vaccine for the COVID-19. So I can take this as an example about the speed. So starting from 2020, when we had the pandemic starting, what happened? And with the speed of the development of the COVID-19 vaccines, what happened afterwards, and what were enablers of this really fast development. And the developments, as you know, are the very special cases. Although these are kind of exceptional ones, we can also have some lessons to learn. Are there anything that we can take for our future practices of drug development? Let me start with something regular or common. So when we have a very fast an accelerated drug development, what benefits can we enjoy? So the drug developments can lead to the success, to have the longevity of the patients. It should go through the pipeline to the patients, and it has to create the markets. Probably there can be many first movers advantages. First of all, let me tell you the first thing on the left side of the screen. So first movers can take the competitiveness as an advantage. The first movers can shape the market, and it is able to have the paradigm presented to the market. In order for the drugs to be used, there should be a connection between the buyers, sellers, and the markets. And also the chains and connections among all these three things can be taken by the first movers. Of course, these treatments should be symbolic, as in a really household brand for the treatments. There can be the linkage of the indications with that product. So the doctors and the clinicians will think of this product and this brand at the first thinking. Therefore, it is only inevitable for these drugs to create a lot of revenues. So it is really literally taking the market. So if we think of this advantage from the perspective of the developers, the fast development saves time, of course, and money. As mentioned, COVID vaccine development was the fastest one in history. So let me take a look at this again. As all of you remember and cannot forget, as a number of year is 2020. In China, if you recall that time, we got this very sudden news. Oh, there's something happening in China, and there is a virus coming up, and it is something called norovirus. That was the February 2019. Only one year after, in 2020, the sequence of the virus was released publicly. And then after a couple of years, between March and April, the pneumonia based on the virus was spreading all across the world very formidably. Many of the shops were closed, and it was the unprecedented situation when we had to have our social distancing. And by summer, very surprisingly, Moderna and Pfizer BioNTech were the two companies who had finished the phase 3 of the clinical trials, or starting the phase 3 of the clinical trials. In the same year, December 12th of 2020, FDA authorized 2 vaccines for the first time in history. All these things happened within 1 year span of 2020. In actuality, if you look at the development situations of the COVID vaccines, I can give you some numbers, what represent these developments. As mentioned before, we had the news of the pneumonia. It was the February 21st of 2020, and then even in February, only 1 year after 2021, they had the clinical development. The number of vaccines for new drugs was 21. By March, they had increased to 125, and by summer, the number was increasing again to 176 drugs. And then also by December, 214 new drugs have been developed for the vaccination. So it was really, truly unprecedented in history. Such a remarkable experience we had was about the development of the vaccines and what would be the significance of these situations compared to that disease. And it was taken from the McKinsey report, and it was the time taken for these developments by phase. So it was the unit of days. So the one small dot means 1 month. So if you think of the processes, let me compare this with the standard cases and the COVID-19 cases. So if we have the drug to be shaped in with some characteristics, and then if they have finished the toxicology studies for the human beings used, and also if you have to have the filings, it will take a couple of years first, 1 or 2 years for the preclinical situations. And then after that, if they are successful, they move on to the next one, which is the phase 1 for the human beings. And in the phase 1, they have to check the safety and immunology, because this is vaccines we are talking about. That will take another 1 year or 2 afterwards. That's for the standard. And then after that, if we are certain that this is okay with the immunology and it is with the efficacy, then we can move on to the next stage or phase, which is for the proof of concept POC. That will take another 2 years, as you know, because that's to communicate with all the researchers in the world. They have to have the peer reviews in order for the drugs to be out in the market because of the characteristics of the vaccinations. This is for prevention of something, prevention against something. Therefore, it should not have any big adverse events. Therefore, the clinical trials should be in large scale, accommodating many people. And so the smallest ones or the fastest ones will take 3 to 4 years for these clinical trials only, because that's taking the most of the time, which is in purple color. And if they are passing through this successfully, then they can go to the filing stage, and then they will be registered in the market, to be marketed, taking another couple of years. They can be reduced a little bit by time, but roughly speaking, they will take just as long as 10 years for one vaccination to be approved and used in the market. However, really, truly shocking thing that happened with the COVID-19 is that everything is taking a couple of months only and a couple of months on each in the stage, and then altogether it took less than 1 year, rather than 10 years. So when we had the vaccines released in the authorization in December, and it must have been only less than 1 year since we started the preclinical studies. How was it possible? We have 4 things to mention here. The first contributing factor is as follows. First of all, this vaccination. For new drugs, there can be many knowledge bases that are needed for this vaccination development. And the knowledge was open and public widely because that was for the survival of the human beings. So regardless of the health concerns or the non-health things, they had to be combined together to share the information and knowledge. Very luckily and fortunately, it was very timely to have the new technologies coming out, which had been accumulated over time to fit into this mechanism of new drug development. Please take a look at the right corner of this screen here. So as mentioned earlier, science is the basis for the new drug development to be started. But when we had the genome sequence for the COVID-19 was established on January 1st, 2020, I would have to say that it was 1 week after the first pneumonia cases were discovered in China. We would call it a luck when we have to say that there was the past experiences compared to the COVID was like SARS and MERS when we had the experiences beforehand. So knowledge was accumulated over these symptoms, and then the sequences were available for the use of all the researchers. So we were able to get into this market very quickly. And most of you must have been vaccinated against the COVID-19, but this kind of vaccination was very different from the previous ones because it is using the mRNA and the vectors. So one thing I can also tell you that it is a lucky thing is because we would not be able to do this if it is based on only the science for that matter. But we were able to get the designation of this part as something that we have to use. We was really ready for use. Only after 4 months of the research, Moderna was the first one to have the production of the first batch. Now, please look at the left corner now. So this is the list of countries that must be very difficult for you to see, but we have the countries here. So which countries are coming from which countries are making those companies. The blue one is the U.S., the down country below the U.S. is China. These two are the biggest countries which had the companies. This kind of thing would not been possible before the COVID-19. The only reason for this to be possible is because we had the commitment and consensus of the humanities to deal with this issue and problem. And then also we had a good level of investment at that time. Because what we need for our vaccination development and the drug development is the ample amount of money. It was possible for us to reach the conclusion and also the market stability within 1 year because of the financing. I can tell you two reasons, two contributing factors for this. The first one is injection of money. So you can invest the money into the companies that are developing these products, and also you can make investment into the brokerages. The brokerage that have been working the most was called CEPI. It's the Coalition for Epidemic Preparedness Innovations. CEPI is an organization that has the filtering the money into the sectors, and when it is designated by WHO as major diseases for the world, the CEPI comes in to have this role. And also, the another thing is that we had pre-purchasing. That became later a kind of political issue or problem down the road, but it was also another factor to contribute to the fast success. So when there is a investor by country, the biggest one, of course, is the US, to be followed by Germany. And the light blue color is public sector. So these countries were making huge investment. So prepayment or pre-purchase was the means used at that time. So the purpose is to have the sharing of the risk taking. So the bottom, the yellow ones, are mostly from the EU, European countries, to be followed by the US and many others down the road. So there had been a very proper level of investment, which was another contributing factor. The third contributing factor was the flexibility. We had our concept of the safety of the human beings when they were injected in vaccines. Having said that, it is also important to have the flexibility of the regulations. That was another factor for the vaccines for COVID-19 successful. I can give you some examples of what happened with these regulations. At the left corner of the screen at the bottom is EMA's cases. They had task force program that was very fast for many vaccine experts to give advice to the vaccine producers. So in order to have some approvals, they should have the package of the certain level of data, because they have to review and they have to discuss those things. But at that time, what they used was a system of rolling review, which means that they had the ongoing process of looking at the data and then having the data on a continuous manner. So the data was injected every time that was available, and that was reviewed immediately after. That made it so fast. And also they had another prime scheme. So prime scheme is the system that can be used for the very fast development, and advice was given to this program by the experts. On the right corner of the screen at the bottom, FDA in the US was also doing very proactive things. Before questions were asked, they published questions for the possible questions. Published answers for the possible questions. So they had the minimum requirement guidelines published, and also they had the criteria for patient selections, designs of the clinical trials, and also the coverages, so that they can be giving permits and approvals when they exceed these kind of guidelines and the criteria. So these regulatory bodies were very proactive. They were giving guidelines beforehand to the public parties before they were asked about their criteria. The fourth contributor factor was the R&D strategy. So I had to explain the operational excellence. This graph seems to be very unfamiliar to you. And if you were engaged in the clinical trials, this is something you might have seen before or might have not seen before. So this is the Janssen and many other companies and also the colored lines means phase 1, 2, and 3. When you have this graph must be very unfamiliar to you because mostly they are linear, sequential line that should look. However, in this case, all the lines are overlapping because the phase 1, 2, and 3 were done at the same time. This was possible because these companies at the left column were taking responsibilities and they were thinking of the shortcuts and the most efficient ones in unison. I'm not going to show you the individual studies, but in the clinical studies, the designs they opted was something very new and unprecedented. And they were also trying to get the fastest and also the most effective ones for the patients. And they had an adaptation scheme over the course of these schemes. And also one example is that they had 3 clinical trials were planned at the same time by one company. And also very interesting points are here, but one thing I can tell you today here is the fact that the new technologies were adopted. So in order for the clinical trials to be finished very early, they have to have a very quick selection of patients. For that matter, they used AI. AI was telling where the patients are, which is the fastest way to get the patients. So they were able to attract the biggest number of patients. Janssen was an example of doing that. They were working with the AI team, and the AI was the facilitator of the clinical trials. So we had these kinds of contributing factors. So it was a great success, and 1 year was taken to have the vaccine development. Let me go back to my first question as a topic, how important it is to have the fast development of the drugs. So if we were not able to fast enough, what would happen? So in this case, in this graph, we had the vaccinations. Some of you must have finished the 2nd or 3rd boosters, and some of you must have finished the boosters. Now, the boosters can be the 1 or 2. So we never expected that this to be so fast. And then you can compare this with the death rate of the people who were not vaccinated. So the unvaccinated people had the linkage of this death rate that is shown here. So up until April this year, the vaccinated people had the difference of the death rate this much. So between the vaccinated and unvaccinated people, their survival ratio was this much different. It means that if you were not vaccinated, the likelihood for you to die is this much high. Of course, this cannot be applied to any disease or every disease we have, but we believe that the collaboration is needed rather than the competition when it is necessary. So when we have the investors, physicians, and the researchers, we should have global cooperation. That's the implication we have. Another lesson we have to take is that we have to be practical in terms of the integration of the powerful tools. Also, we have to have the operational excellence in innovation. We have to be able to execute the operations, and we have to have the allowance of these methods to be used. And also that we have to have very wise use of the investment to take the risk. So for the future development of drugs and of ours, probably these 4 lessons can be taken in the future. So before closing, I'd like to reemphasize my takeaway message. Everybody should be in consensus. It was very important. It was very urgent. So a sense of urgency was the enabler for us to have this fast development because COVID-19 was that much formidable for us. In that case, you can think of other diseases as well. Do you see other diseases that are not urgent in your practices? I don't think so, because we see the oncology. So if you see the graph here, in terms of oncology, if you have the traditional treatment and if you have the new treatment, and you can see that this is the success ratio. This is the likelihood to get into the market. That's the concept you think. Let's interpret this graph if you are a patient. So each dot means each person, each human beings. If it did not have the blue lines, the patient should have followed the red lines. in itself, all these patients must feel urgent with this introduction of the new drugs. Now, I am an expert in the oncology, so the immunotherapy is my expertise as well. So I think I have heard this thing from other speaker today, but we have ipilimumab, which was the drug opening the market of this immunotherapy, and then came down with this, following the line here. All other drugs in the combination of this and the survival ratio had been increased over time with addition of the combination therapies. So the word of urgency should be interpreted differently, not from your perspective, but from the patient's perspective. The earlier the better for the patients when it comes to the treatment of their diseases. The patients can be your friend, your family member, and sometimes yourself. How important is the speed of the drug development? The answer today is very important. I'm sure that you all agree with that. Thank you very much.
Thank you very much, Dr. Kim, for your very, very educational story. Now we move to the next session, next topic, to talk about decentralized clinical trial, environment, and metadata practice. The speaker is Ms. Aparajita Priyadarasini. Sorry about my pronunciation. Dr. Jita has been working in clinical trial and project management long time, and she's a very experienced clinical operations specialist who previously worked for Novartis and then other pharma industry, and then also CRO as well. So today, we all know that decentralized clinical trial management is really important in this important setting through pandemic area. So over to you. Thank you.
Thank you so much. Thanks so much for the introduction. And this is Geeta. You can call me in short Geeta. I'm going to talk about the COVID-19 changes adoption with COVID-19 decentralized trial, which is a very big topic, or I would say clinical, clinical trials industry trend now. And let me just move on to my content. So this is actually all about my content. I'm going to talk about some changes in the clinical trial landscape. Then I would move to elements of DCT, plan and adoption of DCT, as well as metadata practice. So I represent metadata technology. So I think the previous speakers, Dr. Moon talked about the oncology trial and the drug development strategy for oncology, APAC regulations, as well as certain examples where they were doing 8 times protocol amendment. So, and also Mr. Kim talked about innovative technology and safety margin, how technology of inhibitor can help in innovation of drug discovery. And at the same time, Ms. Kim talked about how important the speed of drug development. So I would like to turn around the table and talk about little bit different about the clinical trial, because we talk about drug discovery. Now the implementation of clinical trial, where the clinical operation landscape comes in. So this is actually in general the landscape of change of technology in clinical trial. If I talk about 1863 or 1747, first controlled clinical trial happened. Then 1863, first use of placebo control arm in a rheumatism trial, which is a RA's therapeutic area. Then move on to 1943, where we did the first double-blinded UK cold trial. Then 1947, first element of ethics and guidance, because that's where actually there are principles of ethical clinical trial, how to conduct within the human experiment that was actually discussed. In 1999, that's when the technology came, which is EDC. Medidata is the pioneer of the electronic data capture system. There should be a way how you can really speed up your trial. Then the next one is all about the 2000 came WHO guideline, then I can move on to the different technology, which is basically how to collect the clinician outcome assessment for the quality of life, as well as the consenting done in an electronic way, then so and so for different type of technology. Then in 2020, we started talking about pandemic emergency guidance. That's when all the things actually started. And DCT started introducing by that time. This is just to talk about the dynamic landscape of clinical trial. The green represents the enrollment rate, and the blue represents the rising of the COVID-19. So Medidata did some research during the last fall, 2021 fall, where you can see when the pandemic started, actually, the dip down, actually, the enrollment rate was down. And then later on, it rises at the end of 2020 as well as early 2021. Then from 2021, there is a dip again in the enrollment rate. And then the entire summer of 2021, there is a peak started. This is just to talk about the granularity attitude of country and site level is key to making the best trial. This is just to talk about how the enrollment rate actually went and which other countries actually can be really representing how the pattern is. It's not only because of the pandemic case or the COVID-19 rise of the cases, it is also because of the regulation within the site country as well as the regions. So you can see there are some representative. I can just start with the China. China is green, where actually the pandemic started. So the dip started actually during the enrollment rate within the first stage. Then following red is US, then following Germany as well as UK, which is light blue and the dark blue color. And you can see the South Korea is in yellow. The enrollment rate or the enrollment dip started actually much later than any other country. This is just to talk about elements of DCT. DCT is just not plug and play. DCT is basically the decentralized trial. There are so many considerations you need to have, not only the regulation, but also at the same time, the design of the trial. What are the benefits of DCT? This is basically the definition of DCT and the benefits of DCT. In general, if I talk about a layman term, the DCT is nothing but a customized clinical trial where actually you are making the patient life or the reducing the patient burden and the site burden. At the same time, bringing on the virtual elements to talk to the site, to the patient, and sponsor to the site. And these are some of the benefits you can see: accelerated recruitment, as well as the increased inclusion for generalized result and decreased patient burden, making it easier for the patient, leverage new data source, and decrease site burden. These are some of the benefits you can see. This is just a proposed definition of FDA. FDA says anything you are doing during the clinical trial where the investigational site is not involved. You are doing the virtual elements or bringing the implementation outside of investigational area or the site, that is called DCT. This is just to talk about the Association of Clinical Research Organization, which represents the clinical trial companies as well as the technology company who are majority supporting the entire clinical trial in globe. They actually came up with the entire workflow of DCT. And there are 4 principles which they follow. First is basically bring the trial to the patient. You are not bringing the patient to the trial, you are bringing the trial to the patient. Quality by design about DCT, as well as the guideline for the quality by design. At the same time, risk assessment for the DCT, and the last but not the least is bringing the patient burden down as well as site burden down. This is just to talk about the DCT, which is, as I mentioned, actually it's not only plug and play. So you have to really create otherwise you are going to go, as Dr. Moon mentioned, 8 times, amendment. Just think about 8 times amendment, how much cost and how much time you are spending actually. So before you think about implementing any kind of DCT, the first priority is you need to think about design of your trial. So it is just not plug and play. Every trial cannot become a DCT, especially when we are talking about oncology trial. You have to think about how do you collect the blood? How do you collect the images? How do you analyze the images? So those are the parameters you need to think about. It's just not plug and play. So there are some of the things actually you can consider. And the DCT is basically technology is not of process now. If you are thinking about DCT, it is important to really implement the technology within your protocol. That's how actually you need to think about. Existing process and procedure is also one of the important parameter, because you need to know how capable is your sites, whether they can really, you can go for any type of telemedicine, you can go for remote source document review. But the thing is whether that country and site is open for that. What about GDPR compliance? What about whether they have those facilities or not? The next one is the clinical trial ecosystem. Sponsor, CRO, site, technology, and regulatory agencies. These are also some of the considerations you have to think about. Patient experience and the data source. Data source is also very important. The next topic is plan and adoption of DCT. I would like to highlight some, or I would like to bring some attention on the entire APAC regulation. So if I'm thinking about DCT, the first adopter of the entire DCT is Japan in this region, because Japan has e-consent approved. Japan has been doing the eQua. Japan has been doing DTP as well as the telemedicine. The next one is the biggest market, as Dr. Moon and Dr. Kim and Ms. Kim mentioned, about the South Korea, and then the next one is basically talking about China. China is not ready yet actually to talk about telemedicine, or telemedicine is there, but with the 3 network only, which is basically called Golden Health Network and IMNP, as well as PLA. These are the 3 telemedicine network actually. But if you are thinking about e-consent, the e-signature is still debatable, whether they should take it the e-consent as a proper DCT method or not. Same ways, South Korea, we are thinking about the telemedicine guideline is not very clear. So the telemedicine guideline needs to be really implemented, then you can think about the entire DCT. The secondly, actually, if I talk about Australia, Australia is one of the, country where actually we are taking most of the DCT component, but still there are some components of clinical operations side which is not clear guideline. So this is just the landscape. Then I can talk about building the DCT strategy. How do we build the DCT strategy? These are some of the challenges, so we need to consider. The first one is trial design. As I mentioned, actually, you have to implement the trial design, involve your technology, involve your CRO, involve your regulators while you are talking and producing your entire protocol design. The patient recruitment and engagement, you need to think about the mode of patient recruitment and engagement. Then the navigating the regulatory landscape, which I already spoke about. Engaging the site as a stakeholder. You need to understand what are the difficulties sites are facing before you push them to go for DCT. That's very important. So that's why actually I mentioned the engaging the site as a stakeholder, not as a, you need to do this. Then data source diversification, because the data source diversification is an important thing, because you are going for a multiple country, multi-trial, multi-center. The diversification of data source could be much different actually for site to site. The next one is oversight of monitoring. How do you do the oversight of monitoring? That's also very important. when we are talking about DCT, DCT has two parts. One is the clinical monitoring part as well as the patient centricity. So patient centricity is not the only part of DCT. The clinical operation people, how to manage the data quality. Because I think Ms. Kim and Dr. Moon spoke about the data quality is very important in any clinical trial. Then the summary of the handle DCT challenges is the DCT adoption is rising, technology is part of equation, regulatory landscape, education and change management is critical success. So for example, I can talk about oncology. Oncology trial, people are going towards DCT, then they should actually really think about adaptable trial, because adaptable trial is a trial where actually as and when it is required, you can just add on your components. That's the way to think about how do you really manage the DCT per se. Trusted partner, because not only by looking at the procedure and adoption and your site, you can just go and implement the DCT. You need to have a real partner, the technology partner, real CRO partner, who can understand the entire regulation and how to implement the DCT. Then I would like to jump onto Medidata's DCT practice. There are two components Medidata's DCT practice we consider. The first part is about how to manage the patient and site burden, as well as the second part is how to efficiently collect the data as well as monitor the data. That's the component for clinical operation people. So this is our whole approach of DCT. So we take patient, site, and sponsor as well as CRO. So all the connections should be there. All of them can talk to each other and they can see the data visibility of the data quality. The main thing we can do is site and the patient, the virtual way, or we can take certain components of DCT and we can implement, depending on which type of trial. As I mentioned, every trial can't be DCT. This is just to talk about decentralized tool for patients. This is our patients decentralized trial. So Medidata is the first patient portal. The technology actually is first patient portal, which is actually utilized not only for the recruitment, but also for the entire DCT. I mean, the end-to-end clinical trial can done virtually. So the patient registry, which is basically, you can see here, the registry of patient or the, the report of patient recruitment. This is nothing but a portal where the patient can come and register themselves to participate in the clinical trial. At the same time, the investigator can have the license of this portal to find the exact match of the patient to recruit in the clinical trial. The rest of the things are basically EQA, which is clinician outcome assessment, as well as you can see here e-consent. We do have a telemedicine visit tool, which is completely GDPR compliance as well as regulatory requirements. Then at the same time, you can see there are some wearable devices. So when we are talking about wearable devices, we need to consider the data source. The data source could be humongously different. So the data source we are bringing in and optimizing the entire data in a single data format. So that is called common data format. The common data format can take it and just combine with the patient data and analyze and provide you insightful information. In past, actually, we always used to collect the objective data, but we don't know what to do with that objective data. The biggest challenge is because of the data format. The next slide is all about how does the entire, patient DCT platform is working. Starting from consenting, the patient comes to the consenting. We do have two types of consenting process, either site consenting or remote consenting. Remote consenting can take the approach of taking the televisit module, and then you can do the entire consenting process end to end virtually by having any witness. At the same time, you can go into EQA, as well as we do have a direct-to-patient component. Direct-to-patient is nothing but the patient can receive the IMP at their home. That facility is also available. The next one is all about the clinical operation. How does the clinical operation people can take benefit? So this is decentralized clinical operation, digital oversight. So we take 3 approaches. The first one is the decentralization. The second one is execution and the quality. These are our 3 fundamental requirements to implement any DCT in the clinical operation. This is basically how we work within the clinical operation. So you can see design quality into study. So we take the RBQM approach, which is the requirement of FDA, because there is no way we can do monitoring entire clinical trial actually. Just imagine how much data you are abundant with, and you can't really go through each of the data. So we need to take the RBQM approach. The second one is having a visualized analytics actually, or AI-driven tool, which can find out the anomalies in your data to understand and to maintain your data quality within your clinical trial if you are going for DCT. The third, but not the last, is remote monitoring. How do you make sure when you can't go or any other pandemic happens, how do you make sure that the entire monitoring can be done remotely? So that is also another approach of clinical operation in DCT to maintain. This is just an example, case study actually, I would say. We have done a largest phase 3 clinical trial for a cardiovascular study, and 15,000 patients, 40 sites, and we could found this is an entire DCT approach. That means we haven't taken only certain part of virtual element, we have taken the entire process actually end-to-end virtually. So what was the objective? They wanted to recruit the patient actually faster. They wanted to also get the objective data quality, as well as they want to have the patient recruitment faster. So the solutions they took is the consenting process, the EQA, which is basically quality of life, DTP, electronic data capture system, and the telehealth. The entire process was actually taken, and you can see the collection of we could found 650,000 patient match, which can identify. Out of them, we need to recruit only 15,000 patients. And the objective, as I mentioned, what they got it is platform, the entire platform could really enhance their entire clinical trial recruitment, as well as they can actually got the data quality eventually. Because this is 40 sites, you can imagine everything you are doing remotely. This is just the last key takeaway. As I mentioned, the patient at the center for the data capture. That's the principle of DCT, patient centricity. DCT design transform the site experience, not to give them burden. The second one is regulatory adoption of DCT strategies. So as and when you are going for different countries, the strategy could change, and you have to adapt to that strategy. Patient experience is the best practice because the patient has to be in the center, and you walk around or design your entire trial according to your patient experience. Sponsor and clinical operation oversight principle to practice. I think most of the time when we are talking about DCT, we only talk about site and patient. We never talk about the clinical operation oversight. So I just wanted to conclude my entire presentation by talking about both the approaches or both the fundamental principles are really required in terms of clinical operation as well as patient. So that's all. Thank you so much for having me, and it's an honor to be part of this event. Thank you.
Thank you very much, Jita, for sharing your thought how the DCT should go, and then metadata experience. Now, we are going to move to the last topic, about clinical trial regulation and COVID-driven change in UK and EU. The speaker is Dr. Harry Balanga, who is a chartered biologist and then has long experience in pharma industry for CMC and then other specialty. He is working in EMEA for the quality located in UK. And now we are going to listen to Dr. Balanga's speech. Thank you.
Hello and welcome. My name is Harry Balanga and I'm the Senior Director for Quality for the Clinical Trials Division in the EMEA region for Thermo Fisher Scientific. In today's presentation, I'll be discussing recent developments in clinical trial regulation and the history and implementation of that clinical trial regulation, going through what's changing in terms of expiry dates, etc. Looking at the UK and Great Britain approach. Also looking further afield into the COVID and the new approaches associated with the COVID pandemic, looking at virtual inspections, QP's responsibilities, and having a quick look at decentralized trials before completing a summary. Clinical trial regulation was written historically in part for the express purpose of simplifying the application of clinical trials across the EU. The history has been on the evolution of the national legislation to directives and to greater harmonisation in the regulation. The reasons for the regulation specifically is that each EU member state would have to adopt the legislation straight into EU member state law. Much of the problems at the time were the bureaucracy that was driven from each country changing and adding rules when implementing the directives. This change restructures how clinical trials are legislated and run in the EU. It's been long awaited, given the legislation was passed almost 10 years ago. The delay has mainly been down to the electronic portal readiness and the portal termed CTIS. Critically, it's important that you understand the changes and also the implementation strategy. The implementation is over 3 years. In terms of the applicability, it can be seen here. The regulation came into the force on the 31st of January 2022 and this is the legal basis for clinical trials, and they shifted from the directives to the regulation. Crucially, there is a 12-month period, which is the period we're currently in, where a sponsor can submit under the old legislation, the clinical trial directive, or the new clinical trial regulation. In terms of compliance, there is some complexity. During the next 3 years, up until the 31st of January 2025, the compliance can either be under the old legislation or the new. So depending on the strategy, the sponsor, manufacturers and distributors should be clear which legislation they are acting under. So what's changing? As a reference, there is the old structure and directives referenced here on the left, and presented on the right, you can see the new legislation structures. As I just alluded to, there is a change in the structure. Gone are the old directives and Annex 13, and in place the new regulation, formalising the legislative part and delegated regulations to cover GMPs. Fundamentally, there are changes that affect manufacture, packaging and sponsor submissions. It's important that each sponsor supply chain is subject to a rigorous gap analysis, as there is a raft of new legislation to ensure compliance. A key piece of delegated regulation is 2017-1569, which covers the GMPs for IMPs and is enacted under the clinical trial regulation. Now it's the list of highlighted changes. The new process to sponsor submission is the biggest intended benefit of the clinical trial regulation and streamlines the process of submission. The new process allows a single application procedure via an entry portal under the CTIS. It means a single set of documents and a single authorization procedure against a reporting member state. Included is the ethics approval as well, which means that the timelines are shortened significantly to around 60 calendar days. The clinical trial regulation also introduces other changes, such as changes in documentation retention, formalisation of quality control reference samples, QP certification content detail, and changes in returns and accountability of medicines used within the trial. The 25-year retention of the trial master file has also been included, with the European Medicines Agency referring to a document titled Recommendations on the Content of Trial Master File and Archiving, which is currently being updated, and in the absence of that, you refer to the ICH E6R2 on good clinical practice, which contains the section entitled Essential Documents for Conduct of a Clinical Trial for reference. The clinical trial regulation also brings a change to terminology from non-investigational medicinal products to auxiliary medicinal products. It also is clear that sponsors should use authorised auxiliary medicinal products for their clinical trials. If a sponsor wants to use an unauthorised auxiliary medicinal product in their clinical trial, they must justify their use in the clinical protocol, and its use must be accepted by the regulatory authorities approving. The clinical trial regulation is also clear that cost is not an acceptable justification for the use of unauthorised auxiliary medicinal products. It also states that unauthorised auxiliary medicinal products must have equivalent traceability, returns and accountability arrangements as their authorised IMPs. Greater emphasis on traceability of IMPs and unauthorised auxiliary medicinal products is also at the front, putting the responsibility on the sponsor to ensure that all unauthorised medicines associated with clinical trials are controlled and accounted for throughout their life cycle. There is a specific change around returns and accountability, requiring these arrangements to be detailed in the clinical trial application. To include these details in clinical trial approval and application, sponsors will likely have to determine these arrangements earlier than they currently do in the clinical trial planning process. Finally, and most controversially, Annex 6 of the EU clinical trial regulation changes the clinical label text requirements. Positively, it harmonises clinical label text for clinical trials across all EU member states. The required clinical label text is very similar to Annex 13, but for a few additions. However, the major change that was introduced under the Annex 6 was the requirement for an expiry date to be printed on both the primary and secondary container labels. This allowance provided under the directive in Annex 13 to omit the expiry date from the primary container was not included in Annex 6. This change would have had a significant impact on the packaging of IMPs in EU clinical trials, but I'm happy to say that due to strong lobbying by the industry and industry organisations, the European Commission has completed a public comment and is proposing under new delegated regulation to change the need for the expiry date in Annex 6 to be on the primary pack, which is currently being prepared and published. The industry anticipates that the requirement will not ever come into being, and therefore this is very positive news for sponsors running clinical trials in the EU. However, we are counselled to continue to refer to the European Commission website to understand for updates. In the UK, the MHRA have stated that the UK will not be fully aligned to clinical trial regulation and have a number of suggested changes to clinical legislation under consultation. As the UK has left the European Union, they will no longer be aligned to the same legislation structure as once was. Therefore, in an attempt to improve the smooth running of clinical trials in the UK, a public and industry consultation has been issued, which proposed changes including aligning ethics to regulatory approvals, improving the speed of clinical trial approvals, and crucially, not applying the clinical trial regulation expiry dates to those primary packs. To move on now to the COVID response. COVID has meant that the industry has changed significantly. Among the changes, the most significant being changes in inspections, QP audits, moving towards a more virtual environment. Also changes to decentralized trials and the rapid approval of clinical trial medicines. Remote inspections have become useful in maintaining regulatory oversight during COVID-19, but cannot fully replace on-site inspections. A remote approach could nevertheless become an important complementary inspection tool for regulatory authorities. A review was carried out by the working group of the International Coalition of Medicines Regulatory Authorities in Europe, and in a reflection paper, remote oversight approaches were reviewed in the context of the pandemic, and inspectors assessed the need for inspections and decided on regulatory oversight approach on a case-by-case basis during the pandemic. If it was decided that the remote evaluation was not suitable for regulatory compliance activities, inspections were either postponed until conditions were allowable for the on-site visit, or a hybrid approach was completed, that being one with on-site and remote combined. It's believed that a hybrid approach will continue to be utilised in order to future-proof inspections, and this has been published by various inspection authorities across the world. This could be especially applicable for Asia Pacific based manufacturers where travel constraints continue. The QPs have had a tough time over the COVID-19 pandemic. Not only is it implicit that QPs provide final certification release in the EU, but they do so in an environment where they take responsibility for the supply chain, and they cannot always oversee physically. Many QPs have had to rely on ongoing risk assessments, remote inspections and supporting evidence that would not normally review in order to get through the pandemic. I do believe that regulatory inspections and more remote QP audits will feature in the future, and more electronic means of oversight will be accepted, such as virtual visits and electronic videos and transfers of data. However, it's vitally important to remember the responsiveness of QPs and the responsibility of the QPs have not changed. And so it's important that each request is supported by the sponsor with close relationship to the QP. Decentralised clinical trials have become a necessity during the COVID pandemic. A decentralised trial does not mean one without sites or principal investigators. It's an approach to research that leverages digital technology to treat patients remotely and through hybrid models that often include minimal in-person visits. Since its inception, the most common limitations influencing the ability of the trial to be decentralised are clear regulatory guidance, a country's existing healthcare infrastructure, local culture influencing its acceptability, and the study's ability to run without the level of infrastructure common in traditional trial designs. European countries with regulatory authorities that are more proactive in decentralised trials are also places where active research communities exist. So countries such as Denmark have a positive view on decentralisation. Regulatory bodies in the UK and Netherlands and in Poland are more innovative in their approaches and are more acceptable to decentralised trials also. Switzerland's regulatory body and ethics committee released a position paper on decentralised trials and their adoption over the last year, with some very interesting points, including approaches to hybrid decentralised trials, approaches to good clinical practice in decentralised environment, including digital recruitment and handling of data within a decentralised environment. More guidance has been provided by numerous papers and articles outlining practicalities, such as inclusion of digital technologies, e-consent, telemedicines, wearable devices, electronic clinical outcome assessments, and electronic health records, requiring for the sponsor to be able to support this in that decentralised environment. From a country perspective, Switzerland, as well as the UK and Denmark, saw the sharpest increase in the proportion of trials with a decentralised approach. Clinical trial regulation will help to harmonise the regulatory stances among European countries, with future amendments, perhaps on decentralisation, more likely to be adopted across the region rather than on a country-by-country basis. Further afield, but within the EMEA, in South Africa, recent decentralised clinical trials have really rocketed, predominantly around the COVID-19 and respiratory disease research. There was a big influx of these, albeit simple trial designs, decentralised in nature, partially because of the geographical spread of patients. By contrast, in Asia, single country trials located in China are least likely to involve decentralisation, with fewer than 1 in 100 single country trials having such an approach. However, the adoption of decentralised clinical trials in Asia Pacific countries are increasing, but there are cultural changes that need to be made. Patients in Asia Pacific are more likely to want to see a doctor in person, and so will need to be accepting of new trial designs with less contact. A decentralisation approach that works in one country may not work in another, but decentralised trials need to be because the patient and the trial design are accepting and appropriate. The infrastructure and regulatory guidance provides support, and crucially the sponsor can recruit and run the trial in a novel manner. It's a concept that is proven and appears to be growing in many countries in Europe and across the world. So to summarise, I think it's important to ensure you understand the dates of upcoming changes and changes in clinical trial regulation. It's important that you keep your QPs and regulatory experts close. Perform a gap analysis of the clinical trial regulation and associated regulations and guidance to identify where you need to take actions across your supply chain. Review existing SOPs and make updates to those in compliance with the new regulations. Assess any planned new trials for submission before the 31st of January 2023, determine if they will be submitted under the regulation or under the directive. Assess your existing EU trials to determine if and when they will transfer over from the directive to the regulation. For any trials that have been submitted or transitioned to the regulation, begin planning now for the requirements of the regulation, especially for the labelling requirements if and when they do get changed, but also under the returns and accountability arrangements and auxiliary medicinal product sourcing. The QP role spreads further than you think. It's imperative that the QP understands the manufacturing, testing methods and technologies and the supply chains understood, that they have oversight appropriately to the legislation that they are being applied under. A QP declaration, remember, requires audit information for each QP, and it's important that EU QPs are kept abreast of exactly the changes within that supply chain. Remember, virtual inspections and QP audits may be acceptable, but this will be down to the QP's understanding of that supply chain and the data available to them. I'd like to thank you for listening to my presentation, and I hope to speak to you soon.
Okay, we are going to have 10 minutes of Q&A and until they settle the chair. 저희가 시간이 많진 않지만
Although we have some time constraint, I think that the last speaker spent less than 20 minutes, so we have some time for Q&A. So for the four presentations, or four presenters, if you do have any questions, please do not hesitate to raise them.
만약에 없으시다고 하면 제가
If not, then I would like to ask a question to Dr. Kim Eun-kyung.
빠른 속도로
So you talked about the urgency and rapid speed, sense of urgency, and we have to make rapid decision for clinical trials, but I think that this could be difficult. And you actually give us several case examples. And
사실 그 decision making process를 어떻게
when it comes to decision-making process,
할수 있는지
how
좀 설명해 주시면 어떨까요?
can we ensure the fast and precise decision-making?
감사드리고요.
Thank you for your question.
문학님 선생님께서 워낙 경험이 많으셔서
When Dr. Moon mentioned, and I believe that she is more qualified to answer the questions, and she can give us a guidance. But anyway, in my perspective, decision-making process from the perspective of developers is yes or no. So go or no go. So whether you decide no go or no go, then you can also have a decision about where to go and what the right direction. So it can be a internal strategic
사실은
items that you have to decide upon.
관마다
But in actuality,
식으로 서포트를 하고
in each of the
관만자를
organization, you have to think of your supporter and who is your people on your counterpart.
사실은 어떤
If it is
패스가
not
어떻게
about
가는 것이
strategic
가장
things for your company, you have to find the shortcut and the shortest way to go, eliminating unnecessary things, in my personal opinion. So you have to talk with the authorities very closely to have a decision of these things. So it can be MFDS in Korea, or since you are going to in the U.S. markets, you can talk with the authorities there as well. You should have they have a tool to talk to you from. So you also have to be in use of these tools. Maybe that can be costly, they can be more troublesome, but even if it takes some money and effort, maybe that's the shortest cut that you can think of later. So using all these tools possible in each of the milestone, you have to think of these things and you have to find the best solutions, and that could be the fastest way. What do you think about that, Dr. Moon? Well, I'm in agreement with you. So when it comes to oncology, if we are talking about the time efficient pathway, the RTO, the real-time oncology is the new program that we can use, RTO. So before we have the final decision, say yes or no, we can have the databases that we can refer to, and then NDA packages can be served later down the road. So it is based on the sense of urgency, so it will be very helpful to make a decision very quickly. So any questions from the floor?
예, 없으시면 제가
If not, then I would like to ask a question to Dr. Kim Dae-kwon as well.
여러 가지 굉장히 재미있는
So you gave us very interesting cases.
예를 들어서 AT&T나
For example, like the AT&T and the PI3 kinase, we understand that there are some problems in relation with the metabolism. Now it's a known factor, but when we are in the drug discovery process, and let's say that we produce some target candidates, but if we encounter some unexpected problems, so maybe like the normal cell damage and so on. But would there be any way where we can predict those problems? And in order to do that, what kind of efforts we have to make? So that's a very comprehensive question. Basically, I think that you have to have a biology understanding for target evaluation. And also, in order to develop a selective substance, you have to have some strengths for biomarker development. And so Big Pharma is actually leading that way. And also, the venture startups are more like on the position of catching up. So they can but learn the lessons from the failure of the Big Pharma. So I talked about the GSK for the cooperative inhibitor for the PI3 kinase. In the beginning, they actually failed and they had to stop the clinical trial. But because they revealed all the biology and the mechanism, That's why I think that the other startups were able to have successful clinical trials with the same substance that GSK failed. And I think that these kind of things are something that the small firms could not do. So big farmers, they produce a lot of successful or the failure cases, and small venture startups, I think they can learn the lessons from their cases. I think that's the most realistic approach. Right, so we have to learn the lessons and we have to make efforts to overcome the previous failures.
Question to Jita. You talked about many cases of decentralized clinical trial, and in reality, I don't know whether we can implement DCT at once from A to Z, or at this stage we implement piece by piece and then we combine together later on. So from your own experience or metadata experience, what do you expect DCT would go on in different environment? I know that in Korea, the biggest hurdle is not allowing telemedicine, so I know that. But probably you can share how it would go, either piece by piece or as a whole.
Yeah, sure. That's a very interesting question. And I guess, like, the DCT, as I mentioned during my presentation also, DCT can be really like, depending on the regulatory environment, depending on the country, whether you can take certain part elements of DCT or implement for that particular trial and then go on to like, next country. So for example, China, we can't do anything with the e-consenting because there is a regulation of data privacy, there is a regulation for remote monitoring we can't do. So in that case, actually, but the telemedicine is really established. So we can actually go for telemedicine and as well as the eQua approach, as well as the DTP approach. Same for South Korea, the telemedicine is one of the hurdle. But at the same time, e-consent has been approved and eQua has been there and the DTP can be there. So these are actually, DCT can go with the hybrid model or the entire end-to-end DCT. So it's not necessary that DCT has to be implemented like, the end-to-end. Yeah.
Thank you very much. Actually, I have to share some interesting case. 저 한국말 할게요. 그 어떤 회사가 삼상을 하고 있었는데.
So one company was in the phase 3 clinical trial in the Eastern Europe country, but then there was a Ukraine war breaking out. So they didn't really have the plan for DCT in the first place. But then, as the time for the data closure, lock-in is getting closer, so they had to go for the remote clinical trial, and the CRO did have a lot of experiences, and therefore they were able to have very smooth remote monitoring. And also the patients, they had to receive the assessments in other countries because of the war, and there were a lot of complexities, but somehow they managed them very well. And also the Ms. Kim talked about the urgency and needs. But I think that this kind of urgency can trigger a further development, and they can be turned into opportunities for betterment. And in this regard, I would like to thank you all for your attention and time, and we will conclude the session here.